Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, we have a few announcements to make. The program of the third edition of the Dr. GPCR Summit is taking shape. Our confirmed speakers include Drs. Andrew Tobin, Ryan Roth, Rosie DeWalibi, Karen Gregory, Oliver Hartley, Ines Liebscher, Christelle Manet, and many more. This year, the meeting will be held between October 10th and the 16th. The meeting will be virtual to allow you to join us from anywhere in the world. Live talks will be hosted on Zoom from October 10th to the 15th, and everyone is welcome to participate by presenting a poster or submitting a pre-recorded talk. Similarly to last year, we will have presentation prizes for trainees. We're also trying out a few new things as well. We'll have networking and poster sessions on Wonder. We'll also have a full day dedicated to trainees. If you're a trainee, hurry up and submit your abstract today to give a talk or potentially present a poster. Visit drgpcr.com/summit today. We will also be hosting 3 workshops. We're excited to share that we'll have Dr. Sam Hoare and Dr. Luciana Leo, who will run a workshop on data analysis. Dr. Nicholas Smith will host a workshop on how to preserve data integrity in the lab. And this year, we welcome back the GPCR DB team with Dr. David Gloriam, Dr. Albert Koistra, Gaspar Nandi Sekeres, and Jimmy Caroli. Everyone is welcome to attend the summit. And it's free when you become a Dr. GPCR Ecosystem site member, which is also free. Speaking of the ecosystem, we are excited to share that the Dr. GPCR Ecosystem 2.0 platform is now open. Visit drgpcr.com to explore the ecosystem today. Please note that we are slowly migrating from our old website to our new website. You'll always be able to find us at drgpcr.com, and we thank you for your patience during this transition period. The ecosystem, if you're wondering, is your GPCR-focused virtual playground. Join over 75 of your peers who have already registered as site members. Remember, it's free. You'll also have the option to select a plan and get access to all things Dr. GPCR and much more. You get access to the new podcast episodes with video before they get released to the general public, our new group discussions and forums, our job board, our learning center, where you'll be able to take a course or even prepare and share a course with your colleagues. And last but not least, you'll be able to discover GPCR companies and much more. Take advantage of everything that the new GPCR dedicated online playground has to offer today by visiting drgpcr.com and becoming a site member. Remember, it's free. Once you are a site member, you can also choose to sign up for an ambassador program, which means that you'll get your own dedicated Dr. GPCR ecosystem link. And every time someone subscribes using your link for a year, you'll get compensated. Last but not least, we're also looking for content creators. Subscribe to the ecosystem and start writing your own GPCR-focused content. Share it and show off your talents. You can always reach us at hello@drgpcr.com or visit drgpcr.com to find out more about all our activities. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and today I'm delighted to have with me Hannes Schihada. Hannes, welcome to the podcast. I'm excited to have you on.
Hi, Yamina. Thanks for the invitation. It's a real honor to be a guest on your podcast.
I'm excited to have you on today. And before we hit record, we started talking a little bit. And so let's start at the beginning. Please tell us who you are and what do you do?
Mm-hmm. Yeah, so I grew up in a very small village in the south of Germany, and in the end, I decided after graduating from school, I decided to study pharmacy for different reasons that we might talk about later. And there was several twists and turns throughout my career, and I always changed my plan, and then in the end decided to do something else than what I had thought of doing after finishing this step. And that's how I bumped into doing GPCR research. I'm currently doing my second postdoc in the lab of Peter Kolb in Marburg in Germany. Before that, so after my studies of pharmacy, I did my PhD studies in the lab of Martin Lohse in Würzburg Then I moved to Stockholm in Sweden at the Karolinska Institute, where I joined the lab of Gunnar Schulte working on class F GPCR, so mainly frizzles. And yeah, today, as I said, I'm back in Germany, reunited with my wife after 2.5 years of a distance relationship while I was in Sweden. And I'm trying to continue here, and I'm seeking, of course, as many others do in this competitive field, trying to find a permanent position which would allow me to do GPCR research.
Awesome, awesome. I have a ton of questions already. So, why pharmacy? Why did you get into pharmacy at all?
Yeah. Well, I should mention, well, none of my parents is a scientist, none of them is a pharmacist. My mother is a teacher, and my father, he studied chemistry. So he came to the GDR from Israel. He's Arabic, and he came to the GDR with a stipend to study chemistry. And he was really interested in chemistry. And while I was at high school, without having chemistry lessons at school yet, we started talking about chemistry already at lunch or whatever. So my father started asking questions like, "Yeah, do you know how a battery works?" or something like that. And then, of course, as soon as I had chemistry classes at school, these discussions over lunch or over dinner got more and more intense. So my mother and my younger brother were bored by these conversations, I guess. But these conversations actually sparked my interest into natural sciences and chemistry more specifically. But after I finished school, I thought what I would like to do. And my father always said, although you're interested in it, don't study chemistry because it's a hard way. And I mean, as I said, I was interested in all the natural sciences, biology, physics, but I was also always intrigued about medicine. I had no understanding of it, of course, but I was always intrigued and wondered how actually drugs do what they do. Why can you take a pill of aspirin and it knows how to inhibit this particular enzyme that kills your pain? And so I thought maybe pharmacy is a good combination of all these different subjects. And but the original plan was at that time that I study pharmacy to, in the end, after these 5 years of studies, to own my own pharmacy and manage my own pharmacy. So I would be my own boss, which was a very attractive idea at that point for me. And yeah, so that's the reason why I started studying pharmacy.
It's interesting since you mentioned about your background and having an Arabic background. I think it's very cultural as well. This being independent and owning your own thing, and also medicine. I was surprised that you weren't pushed towards medicine, because typically, knowing my own background, it's either you become a lawyer, potentially a pharmacist, a dentist, or a doctor, and that's about it when it comes to
Yeah.
to family pushed backgrounds
Yeah, that's a good point, and I think my father was trying to push me, or well, I'm sure he was definitely trying to push me towards medicine. But I think the reason why I then chose pharmacy instead, pharmacy was basically the second option, which was also okay for him. But I think I was too afraid of these pathology classes where you would dissect the head of a pig or whatever, and or work with dead bodies. This was I could not see myself in this lab course, to be honest.
Yeah, so you took the next best thing, which is really thinking about drugs. And then you've completed your pharmacy degree, and then how did you get into research?
Yeah. So at the very end of these studies, the last year of these 5 year studies, you do something that is in like literally translated from German, like the practical year, which is two times 6 month internship where you have to be supervised by a pharmacist, and 6 months. So one part of that should be done in a pharmacy, like in a typical public pharmacy in Germany, and the other one is free to you to choose whether you want to go to industry, whether you want to do it in a research environment, or even in the clinics, if you find a pharmacist who would supervise you there. And I decided to go to Hoffmann La Roche, to the company that has its lab, its headquarter in Basel, in Switzerland. So I went there and did an internship at the drug production unit for solid drugs. And I really enjoyed it there. So it was a lot of quality assurance, quality controls, a lot of desk work, of course, but I really enjoyed. I had nice colleagues. I was interested in the things they did at work, and of course, with the pharmaceutical background, it was also from the very beginning there was quite some understanding how drugs are developed and how you assess risks if something fails during the production line, for instance, risk to the patient or whatever. So this was a very nice time, these 6 months in Basel. And after that, I thought, okay, I definitely want to pursue an industry career in pharmaceutical industry. But I also learned during these 6 months in at Hoffmann La Roche that having a PhD would definitely boost your career and would make it easier to get to higher positions or even to find a position there in big pharma. And that's why after this practical year and the final exam of pharmacy, then I was looking for PhD positions. And what I was interested in most during the entire studies was always pharmacology. So there were very nice lectures by, for instance, Armin Buschauer, who is a medicinal chemist doing research on GPCRs, who did research on GPCRs. Unfortunately, he passed away a couple of years ago, way too early, but he was very passionate about GPCRs, and I found them very interesting, but especially this connection between the chemistry of the drug and its action and why it sits in a specific pocket and why a drug targets this protein but not another protein. This I found always very interesting, but it was not only about GPCRs in particular. I was just trying to find any position in pharmacology to do my PhD. And yeah, then there was a new consortium starting in organized by different universities in Bavaria, so in the south of Germany. And they organized an interview day in hot summer in Würzburg, which is a very nice town, by the way, and surrounded by vineyards and hills. So it gets very hot there. And they organized an interview day. And these were labs led by, for instance, Martin Lohse, but also Davide Calibiro, Carsten Hoffmann, Peter Gmeiner. So at that point, I didn't know, but big people from the GPCR field. And they had, I think, something like 10 PhD positions. And they invited, I don't know how many different applicants to this interview day. And everyone was supposed to present something that he did, like an own project that he was working on during his studies. And I was there with these studies, like pharmacy, that is very school-like. You get your time schedule at the very beginning of the semester. I was sitting there and thought, okay, well, what could I present? There's no bachelor's thesis, no master's thesis. So in the end, I picked a lab course that I did with 3 of my colleagues during my studies on pharmaceutical technology, like you change the recipe of a tablet and check how it affects the break resistance of this tablet. So very, very far from pharmacology and from medicinal chemistry. But that's what I presented there. And I don't know for what reason, but for whatever reason, I was then offered a position in Martin Lohse's lab as a PhD student in pharmacology, of GPCR pharmacology.
That's such an interesting way. And I'm sorry, by the way, I got signed out of Zoom and I had to sign back in to make sure that we're recording, hence my typing here. Sometimes these things happen. And it's so interesting that, you decided, I understand that you decided to do a PhD because the thought was, okay, if I get a PhD, then I have a better chance to go back to industry at a higher position. Where did the idea of choosing that project to present come from? The presentation that you made about changing the formula of a drug. And do you think, have you ever asked Martin what caught his eye or what he liked about that presentation?
Yeah, unfortunately, I haven't done that. We're still in close contact. I should definitely do that and ask him why I was one of the ones who were then selected and offered a position. But I don't really know, to be honest. I chose this topic because I had no other choice. Basically, as I said, in pharmacy studies, there's only very few internships that you could do.
Yeah.
I could have mentioned there was one more internship that I did with Novartis, but this was like only for 4 weeks, and what I did there was mainly like a literature research and summary of the current literature on multiple sclerosis, which I found also very interesting, but there was nothing really to present. So this was the only option. I remember though from this interview day that I was there after these 20 minutes talk, which was, by the way, the first talk for me in English. So I felt very uncomfortable. But in the end, they asked me, yeah, why did you change this? And what is the current knowledge? And I could not really tell because it was just about, well, something that I did as a typical curriculum of these studies.
Yeah.
But Martin Lose then asked me after the presentation in the discussion whether I would be more interested on the protein side or the chemistry side if I think about GPCRs. I had no clue at that point what he means. Yes, chemistry and protein. Yes, I knew what he means, but I didn't know how it would shape my career in the future. What he was basically asking was would you be interested more in the synthesis part, or really looking at the biological function of these receptors? And I mean, I could not think about it that fast, but I thought I should give an answer very quickly and very confidently. I just said proteins, definitely proteins. And this is how, or what shaped my trajectory as well, just by accident, basically. And in the evening, sorry.
I'm sorry. No, please go ahead.
Yeah, and in the evening, so at the end of this interview day, we had a very nice dinner, all the applicants who traveled to Würzburg. And there then Martin Lose approached me and said, look, I saw from your CV that you were interested, or that you did internships in industry, and is this something that you could imagine after your PhD as well? And I said yes. And then he said that there's this project where they are basically trying to use a technique that I now know is Fred and Brett, that you can use to create conformational sensors of GPCRs. That they want to use this technology that they have developed already in the early 2000s with seminal work by Jean-Pierre Villadaga and Moritz Brünemann, that they want to harvest this technique to develop assays for high throughput screening of GPCRs using this conformational readout. And the idea would have been that if these new sensor generation would be sensitive enough that one could even think about a startup idea. And that also sparked my interest. I thought, okay, this sounds a bit like applied science and going towards the industry direction.
Yeah.
And that's why I definitely wanted to take this position and took it in the end.
That's amazing. The next question I had for you, you mentioned, first hearing about GPCRs in the lecture and that lecture. Can you remember when was the moment when you were like, okay, these proteins are super interesting and I definitely want to work on them? Or it kind of fell into your lap and you ended up falling in love along the way?
Yeah, I think it was more the second option, like falling in love the longer way, because I mean, from the lectures, especially from Armin Buschauer, I remember these spaghetti plots that I know now as the crystal structure, because I studied 2009 to 2014, and then in the middle of that, the first crystal structure of the beta-2 receptor was published by Brian Kubilka's lab. And so he was, of course, very passionate about it, but I, sitting in the lecture hall, did not really grasp how this is. Of course, I found this interesting, but I didn't really know how relevant this is. And so I always thought about, before I started my PhD in GPCR pharmacology, I thought of GPCRs as always like remembering these spaghetti diagrams from Armin Buschauer's lecture, which was very funny. But then when I was offered the position by Martin Lose, I read more into it and I realized how amazing drug targets they are and how many receptors there still are that are untapped and are not exploited for drug discovery. So this was maybe the second step where I thought, okay, this is a very cool topic. And then, during my PhD over the years, I read more and more about the biological function and the physiological aspects they're involved in, and especially how complex they are. That one receptor can do this thing with one ligand and a very different thing with another ligand, or receptors that look so similar do very, very different things, or even the same receptor doing different things depending on the cell line or the tissue. So it was more a development over time.
That's actually quite cool, and I think you're a unique guest in the sense that you've completed a separate degree, you've had some industry experience, and then decided to move on and do a PhD, which I think is really admirable, and also gave you a completely different look at the PhD, knowing that you had a goal, which has changed over time, but you did have a goal and you had prior experience. So you knew why you wanted to do a PhD, which is, to get that knowledge and then go back to industry. And then at the end of your PhD, what took you to Gunnar Schulte's lab?
Yeah, so I think this decision or the change occurred quite in the middle of my PhD. So when I decided to not seek for industry positions, but to do or to try to stay in academia at least for another postdoc. And I think one key event in that process was at the beginning very unfortunate for me. It was that Martin Lohse decided to move his lab to Berlin because he became the head of the Max Delbrück Center, so a big research campus in Berlin.
Yeah.
And the whole lab moved with him, with two exceptions, which was the postdoc who was supervising me, Isabella Mayer-Lau, who is now a group leader in Nottingham. And because I was closely supervised by her, I also decided to stay in Würzburg at that time. But I think a lot of funding was still fixed to Würzburg. And this is why for the last 2 years or 1.5 years, there was a lot of money to buy consumables and try out different directions of research, but only for these two people who decided to stay in Martin Lohse's lab in Würzburg. And then I realized how much fun it actually is to be free in what you do. Of course, I was always using these resources to do things that are part of my project, but it also gave me the freedom to explore different things around these particular receptors. And this was very, very inspiring, which led me to say, okay, I want to do I want to do a postdoc. And well, that was then, I think, summer 2018 when that decision was made. And I was looking for different postdoc opportunities. And I saw just by accident of this GPCR consortium page on Facebook that is, I think, managed by Thierry Hébert.
Yes.
That he posted a job offer of Gunnar Schulte. Later, Gunnar Schulte told me that this was something like 2 years old, and just by accident, it came up in my timeline, apparently. But I thought, okay, Gunnar Schulte, I heard a talk of him a couple of years ago when he was visiting Würzburg. It was very interesting, and frizzles are also very exotic. And I should have mentioned that, like, my PhD was developing these conformational sensors, but I worked exclusively on class A and class B GPCR, so receptors that we better understand than maybe frizzles, I would say. And I thought, okay, maybe I should take this knowledge to less understood receptors, and maybe I could use this expertise that I acquired over the course of my PhD and apply it to frizzles. And that was the initial idea. And I contacted Gunnar Schulte. We had several meetings, and he was interested in me joining. And yeah, luckily I got then a fellowship and decided to move to Stockholm.
Wow, a big decision, knowing that you ended up living apart from your wife for that time. You mentioned working on class A and class B GPCRs during your PhD, and then moving on to frizzles. And I have to ask this: do you have a favorite GPCR?
I think there are several receptors that I like. There are for example, just control receptors that I use for my experiments when I, for instance, try out a new assay for GQ coupled receptors or G12, G13 coupled receptors. And I like working with, for example, the M3 muscarinic receptor. I like working with the thromboxane A2 receptor, the histamine H3 receptor. But this is more like control receptors. Of course, I have to mention frizzles here because I find them so fascinating because of the way that they work. They are very similar to GPCR in some aspects, but also very different from them in other aspects. But what I am working on right now, or what I started already working on several years ago at the end of my PhD, is a cluster of orphan class A GPCRs. So if I would have to call one or a cluster of GPCRs, then I would say GPR3, GPR6, and 12, which are homology, like very, very similar in homology.
Interesting. Well, I always ask this, and I get different answers depending on who I'm talking to. Some, for example, Michel Bouvier said he doesn't have a favorite GPCR. He thinks of GPCRs as great tools to better understand, as a receptor to better understand the biology of a class or the biology of the family. We were talking about Andrew Tobin before we hit record, and he actually has a phosphorylation motif on a GPCR as a favorite. So it's really, I've been thinking about it. I'm thinking about it, and I need to change the question a little bit because there is no right or wrong answer. It's really a preference. You mentioned frizzles, and you mentioned that frizzles have commonalities with Class A or with regular, what we call a regular GPCR, but they also have differences. What would be those, differences, and why do you think that is really cool to study?
Yeah, well, I think what we, especially then in the last years, but the idea has been around in the field and in Gunnar Schulte's lab before, or several years before that, of course, as well, is that these receptors have at least two different ways of functioning. And what is, of course, very different from the rest of the GPCR field is this mode of activation and signaling that is actually promoted by Wnt binding to these receptors and then cross-linking them with what are called Wnt co-receptors, such as LRP5 and 6. So Wnt binding then cross-links frizzles with LRP5 and 6, and this then leads to mutual phosphorylation at the C termini, so at the intracellular sites of frizzles, but also of LRPs, which always reminded me a bit of receptor tyrosine kinases, right? That also dimerize, hetero or homodimerize, and then there's phosphorylation that then leads to the recruitment of signaling proteins. And it's similar for frizzles, that then these cross-linked complexes that are also called signalosomes, that this then leads to the recruitment of a so-called destruction complex. One of the crucial components there is disheveled, which then, like further in the cell, leads to this, what is often referred to as the canonical signaling pathway, a term that is that one should avoid working in Gunnar Schulte's lab, but because it just doesn't like, which I understand, this term canonical and uncanonical, but which leads to this beta-catenin dependent signaling pathway. So this is very different from other receptors. I think there's only very, very few studies or reports in the literature of other receptors, non-class F receptors, that do a similar thing. For example, there's one study on the PTH1 receptor from 2010, I think, that reported on also disheveled being recruited or interacting with the PTH1 receptor upon agonist stimulation and leading to beta-catenin, but I don't know whether this has been reported by others later as well. So frizzles are very different in that. But the idea that has been around is that for very long in the Schulte lab is that frizzles, very similar to class A, class B, class C GPCRs, do undergo conformational changes, and then, like in line with the ternary complex model, that these conformational changes would then lead to G protein coupling or arrestin coupling, GRK coupling to these receptors. And this is what we have, or that I have been studying as well with these conformational sensors for frizzles. And I think we made a good contribution and showed that indeed these receptors are able to undergo conformational changes upon agonist stimulation, and that they can behave similarly to class A, B, C GPCRs.
That is so interesting. Again, sorry, I got thrown out again out of Zoom. Um, so the question I had, this whole Zoom thing just made me lose my question. You mentioned that in Martin Loz's lab, you developed a lot of FRET and BRET-based conformational sensors, and you took that with you, that toolbox basically with you, into your postdoc to apply it to the frizzled. How difficult was it to work with, to translate that into the frizzled world?
Very difficult. It didn't work. So during my PhD, I developed basically, I took sensors that were already established for class A GPCRs, for example, the alpha-2A adrenergic receptor, FRET sensors that were based on CFP, YFP that we knew at that time work very well under the microscope, but they are not sensitive enough to put them into a microtiter plate and then where you have measure basically an average of whatever 80,000 cells in a well. So you need to have a very robust assay and a very robust sensor. And my task during the PhD was to try different combinations of FRET and BRET partners to find the most sensitive FRET design for such kind of biosensors. And what we found out was most sensitive, at least for these three receptors that we studied, was a combination of nanoluciferase with a red fluorescent dye that you would incorporate into the receptor using halotech technology. So also in my DFG fellowship application, I wrote that I want to use this system for class F receptors. I just want to put these tags at the C terminus and within the different parts of the intracellular loop 3 of frizzles and see how or whether these fusion proteins would then report on conformational changes. But when I did that, starting with frizzled 4, which I used as a model receptor, I could not detect any significant changes in BRET upon stimulation with WINS. I don't really know why. I think the surface expression, it's often the surface expression of these fusion proteins that is just screwed up and doesn't allow these proteins then to be trafficked to the membrane. But the expression at the surface was okay. I just don't know why it didn't work. Maybe because frizzles just act differently in how the conformational changes is basically running through the receptor. So I was very frustrated in my first months when this technique that all my fellowship application was built on failed. But then I looked into the literature and I found this technology that has been published a couple of years earlier, or in 2018, for GPCRs or for GPCR conformational sensors that is based on the circularly permuted green fluorescent protein that you would insert in the third loop of your receptor, in the third intracellular loop of the receptor. And then conformational changes of the receptor would lead to conformational changes of CpGFP of this permutated fluorescent protein, and then lead to a change in fluorescence intensity. So that was the backup that I found, and I was very happy and but also lucky, of course, that it worked out for frizzles.
How did you deal with the disappointment? I think this is an important thing to hear about when you're a PhD student or a postdoc, because we all know that 90% of the experiments that we run just end up not working.
Yeah, that's a very good question, and I have to say that it was a very hard time for me. This was, I mean, imagine I just moved to Stockholm. My wife, my whole family stayed here in Germany, and I had this plan in mind that I thought is good. I started that, and I saw that it just doesn't work out, and I thought, oh, what the hell? Now I moved here. My whole career is basically dependent on my success here as for my first postdoc. And yeah, what did I do? I talked a lot to Gunnar Schulte. Just any idea that I had for projects, also alternative projects, not only for developing conformational frizzled sensors, but any projects that I had, I talked to him, and he always had an open door and listened to me and encouraged me in trying out different things. And yeah, in the end, it's trying out as many options that you find. And if one of them is successful, just follow this path and continue. And you always need, of course, strong support from family, friends, your partner, and you just need to feel comfortable in your environment. And that's what I definitely did in Stockholm. And I had a very great mentor with Gunnar Schulte, as I said, who was always very encouraging and said, yeah, sounds like a good idea. This sounds less feasible, but if you want to do that, do it. Just try out. You have to make your own experiences. We tried that, I don't know, 10 years ago, and it didn't work. But he just gave me the freedom to explore, and that helped a lot to deal with my frustration from the first months.
I think you made a couple of great points. One is always have a backup or two, and be open-minded as well, and have the support or seek the advice and support of your mentor, of your peers and family, obviously in different types of contexts, but having someone to talk through your experiments and to talk through your failures is very important because it gives you the ability to verbalize what's in your head. And sometimes, and again, I remember when I was talking through my difficulties, just saying it out loud, I realized, oh, I should try this, or oh, this wasn't the right path to go for. I think communication becomes key to hear your own thoughts as well.
Yeah, and as you said, I mean, also get inspiration from other people, and or inspiration just from other papers. In that case, it was just that I read this paper. I think that was published in Science from a group in San Francisco, and they developed these CPGFP based sensors for dopamine receptors. And I thought, okay, for whatever reason, the bread approach didn't work, but why not trying this for results? So you need to read, you need also to get connected with other people in the field who maybe went through all of this already. Yeah. And just, as you say, be open-minded.
Yeah, sometimes it's disappointing, when something doesn't work, but I feel like science just gives us all these slaps back and forth, and the whole thing is just a trial through fire to see if you're going to get through that. And it is what it is, and you just have to persevere and go from, I say, dopamine rush to another dopamine rush when things finally start working. And typically you're alone, it's 10 p.m. and nobody's around in the lab, but you're dancing because your Western blot work or whatever data you're looking at looks amazing.
Yeah. And I think, well, something that you cannot really control, but being passionate about what you do is very important when, as you say, failures slap your face. Then it's very important that you know why you were doing this and why you were trying this out, because you want to understand how this receptor works, for instance. And that's definitely one thing. Try to do what you're interested in, and not just do something that is on vogue at that time, but what really strives you the most.
That's also, I think, great advice. And we've heard it multiple times from different guests, is passion drives through these moments where things just don't work out the way you thought they would. And being able to spend time in different labs and being able to have different experiences, whether they're positive or negative, it still adds to that arsenal. It builds resilience, it builds confidence, gives you patience, and then knowing what you really, really like allows you to get through these tough moments.
Yeah, definitely.
We've all had dry spells. I mean, even Bob Lefkowitz's experiments failed for a year before anything started to work. So I think trainees, we need to keep that in mind in general, not only in the lab, but in science and in life as well. Yeah,
definitely. And I often say that to PhD students, for instance, that the PhD, these 3 to 4 years or even longer of a PhD, this is not about publishing a paper every year. It's about learning, and then in the end, if you're lucky, you can harvest and publish 1 paper or 2 papers or whatever. But the real focus of the PhD time should be learning, and you can learn the most from your failures. So failures are actually good for that process.
Yeah. Yeah, definitely, definitely. And depending on which country you're doing your PhD in order to graduate, sometimes you still need to publish. Um, and that can be very stressful, but I think following your passion and learning from your failures is going to lead you to that publication for sure.
Yeah. Yeah.
And sometimes you're just not lucky and you have a project that doesn't pan out. And I think it's also important to fail fast so that very quickly you can pivot and go into a different direction.
Yeah, definitely. So that's especially what I tried during when I started there in Stockholm the first months. Of course, I had this like a core project with the conformational sensors, but on the side, evenings, sometimes weekends, I was also trying to like go other directions, use other biosensors that I knew that worked, cyclic AMP sensors, G protein sensors, whatever, that I knew that worked for class A and B GPCRs, to use them in combination with frizzles to see whether this would give a nice backup story. And years later, I could sometimes use the same data that I acquired there, but that at some point then was buried in the drawer because it did not give a whole picture. But we could then add this to another part of the project, and it was not for nothing.
Yeah, and it's also important to know what doesn't work. I love when you mentioned that you had these conversations with Gunnar, and he said, "Well, we've tried this 10 years ago, it didn't work, but go ahead." Which I think is also important to know that it's been tried out and it didn't work back then, but maybe back then the technology wasn't as developed, or sometimes there is these people who have these magic hands that make things work, and you wonder how did I did the same thing? How come it works in your hands and not in mine? And then it turns out that it's a little tweak that they're doing and that you haven't been doing. So I think it's really important to keep an open mind.
Yeah, definitely. And that's a good example for GPCR sensors as well. Sometimes it's just one or two amino acids that you need to shift your tag, and then it makes it to the surface and reports on the dynamics of the receptor. It's very weird sometimes.
Yes, and there is no logical explanation to it, or at least we're not aware of that. There is that luck component as well. And I wish sometimes we'd had this magnifying glass to kind of look at the receptor and be like, okay, what's your problem? How can I help you to signal, help me, and
Yeah.
get that. So after your postdoc in Stockholm, what drove you to do a second postdoc? And it's clear that you wanted to go home, obviously, to your family, to your wife. But how did you pick your second postdoc?
That was basically because of a collaboration that was already ongoing with Peter Kolb. So I should mention that, as I said before, at the end of my PhD in Martin Lose's lab, there was a lot of freedom and resources that I could use to explore different directions. And one of the things that I tried and that we haven't published until now is that at that time, when I saw these conformational sensors working well for a couple of class A and class B GPCRs, I thought, okay, why not use the remaining time that I have here before I go to Stockholm to try to clone and validate a sensor for a receptor that is, like, from a therapeutic perspective more interesting, or also from a biological perspective more interesting. And so I was very naive in picking a new receptor that I would like to work on, but I thought, okay, let's go for an orphan receptor, because these are obviously very poorly understood, and they could also become therapeutically more relevant if we know what these receptors do. And I remember, like, going through the list of orphans. I don't remember which review I found, but I tried to find a review on orphan receptors and what we know about them and what not. And I mean, it started with GPR1 and ended I don't know where. And there was a table in there that said whether synthetic ligands are available or not. And this is very crucial if you want to design a conformational sensor for your receptor of interest, and you want to validate whether it reports on conformational change or not. If you don't have a ligand at hand, then this is very hard to do. And so I ended up with GPR3. So I didn't go down further the list than GPR3, and I saw that there's a synthetic inverse agonist for this receptor, and that's how I started. I cloned this fusion construct, and for some reason it worked out. And Yeah, this is a project that I'm focusing on right now with my second postdoc here in Peter Kolb's lab, because we use this sensor, or we're trying to use this sensor to screen for drugs, or to screen for new ligands, I should say, not drugs, but for new synthetic ligands for this receptor, because we think that this conformational readout is, well, because it's so proximal, you don't need to know which signaling pathway is activated by your receptor, which is sometimes hard, or often very hard for orphan receptors to say. But you can just follow the conformational change. But instead of screening in the wet lab 1 million of compounds, we thought, Martin Lose and I, that it would be very good to team up with computational people to run virtual screening. And this is how our collaboration started with Peter Kolb while I was still in Stockholm. And we had this contact, and I liked to talk with him about the project. And I thought the next step, it would be good to join his lab here in Marburg to continue with what has been running on the side for GPR3 over the years, and focus on that and try to discover new ligands for this receptor using computational techniques in combination with this conformational biosensor.
Love it. What does GPR3 do in real life?
Yeah, there's not much known about it, I have to say. So if you just enter GPR3 on PubMed, there is not too many publications that you will find. But so it's definitely part of a cluster. So there's GPR6 and GPR12 that are very closely related to GPR3. And these three receptors, GPR6 and GPR12 in particular, show very high expression in the central nervous system. GPR3 seems to be like a bit broader distributed across human tissue. You can also find it in the reproductive system, in adipocytes, for instance, as well. And yeah, what does it do? There are some very intriguing papers, especially for GPR3, that links the function of this receptor to Alzheimer's disease. Mm-hmm. So there was a publication, I think from 2012, that said that GPR3 recruiting and interacting with beta-arrestin, that this complex GPR3 with beta-arrestin will stimulate the enzymatic activity of the gamma secretase, which basically leads to the production of amyloid beta. And then also to these amyloid beta plaques that you also find in patients, often find in patients suffering from Alzheimer's disease. Similar to that, GPR6 is associated with Parkinson's disease. And there is now a synthetic inverse agonist for GPR6 that is more or less selective for GPR6. So I think around 100-fold selective for GPR6 over GPR3 and 12. That inhibits this receptor and is now undergoing clinical trials for Parkinson's disease. And well, another thing that should be mentioned for GPR3 is also a very big paper that came out, I think, summer last year, that linked the function of this receptor and its constitutive activity, especially so GPR3, 6, and 12, seem to be, or are constitutively coupled to GS and activate the whole GS cyclic AMP cascade in cells. And this study had shown that if adipocytes are exposed to cold, then the expression level of this receptor goes up, and this drives then, because of its constitutive activity, thermogenic programs, which could be very beneficial in terms of metabolic diseases. So this is also something that I find very interesting. If we would, for instance, discover agonists for this receptor, this could be used to treat metabolic diseases, whereas inverse agonists could be used as drugs or ligands against Alzheimer's disease if we find those for GPR3.
Yeah. I think it's really interesting. You were talking, and in my mind, I was like, oh my God, there are still GPCRs that do amazing things in the body, and we haven't discovered them. So there is so much more to be done.
Yeah, definitely. Yeah.
Do you think this biosensor that you're looking at to screen for GPR3 ligands can be translated to other orphan GPCRs?
In general, yes, but it would require a lot of trial and error, and there might still be some receptors that just don't work, as I have experienced with frizzles. For some receptors, you just don't know why they don't work. But this design for GPR3 is basically the same design that we have used for during my PhD for the alpha-2, for the beta-2, and the PTH1 receptor, also for the histamine X3 receptor. So there are a number of receptors where this design seems to work well. So I think it should also be transferable to other orphan receptors, but it's always a lot of trial and error.
Why? One other question: why you mentioned that GPR3, 6, and 12 have this high level of homology? Why only focus on 3 and not do 6 and 12 at the same time?
Yeah. That's because of the tools that we have.
Okay.
We have this conformational sensor now for GPR3, and my strategy would be to first focus on GPR3, but of course, anything that we would find, any ligand that we would discover to be active on GPR3 would then, on the next step, be validated and tested on GPR6 and 12 as well. But for the screening, we thought it's good enough to go with one conformational sensor and not use GPR6 and GPR12 sensors in parallel, because they're so similar. Like we're talking about 60% sequence homology or similarity that I guess ligands that we would find for GPR3 could likely also target these other two receptors.
Yeah. So it might be redundant to do it for all three, but focus on one and then see whatever hits you have and test those on 6 and 12.
Yeah, exactly.
Makes sense. We've talked a little bit about, your career decisions and how those changed and what led you from talking at lunch or at dinner with your dad about chemistry to where you are today, and a little bit about how you've picked your next step. Any other advice you would have for junior scientists who might want to contribute to the field or to science in general?
Yeah, well, I think as I could experience myself, freedom is freedom to explore what you're interested in is very important for your development, I think. So if you could choose a lab or a supervisor that can tell you that you would have a lot of scientific freedom to explore different directions, this is definitely a pro, I would say. So this is advice I would give to junior researchers to really try to find these groups and these PIs that would give them a lot of scientific freedom. On the other hand, what I mentioned before, one should, I think, follow his or her own interests. Don't pick a certain research topic just because it's on vogue at that time, but pick it because you are interested in that. And try to get a big overview of your field by, for instance, listening to the Dr. GPCR podcast, reading books. I love the book of Bob Lefkowitz because it gives you such a nice historic overview as well. Not only, of course, insights into his personal life, but also the history of GPCR research, which can be very inspiring and can open up your eyes when you have a new challenge in your career. And join conferences, get inspiration from other people. These are the advices that I would give.
I think that's amazing advice, and I think it's important as well. The one important point that you made is really being able to be free and work on projects that you're passionate about and not restrict yourself to the latest trend is very important. It is sometimes difficult, especially when you're going for funding. If you're not trendy enough, then it might be risky. But then again, that's why you should have multiple projects and try and design that, because I feel like I can think of Brian Kobilka's projects, and he was mentioning this at I think it was right after the Nobel or right before. It was after, where he was running low on funding, and the purification columns were in his office because he didn't get funding for that project, those projects, and he had to work on something else. But then he persevered and he obviously succeeded. but that's also important to keep in mind, kind of navigating what's on vogue and choosing a project that might have a higher chance of being funded, but at the same time, don't necessarily focus on what's trendy. Really follow your passion.
Yeah, definitely, but still have the big picture or the big overview in mind. I think,
yes,
although your project might be focused on one particular receptor or even a particular aspect of this receptor, and other topics in the GPCR field might not be relevant for this particular project of yours right now, but it could really help you later to when you have to write your own fellowship application or grant application or whatever, or if you just try to understand your own data, that there might be another aspect of this receptor that could come into play in this particular experiment that you were running and that you just don't understand at that moment.
And again, you make a great point about having this overview of the field. It's important because it might give you inspiration. You might find a technique, you might find a biosensor that was used for the dopamine receptor that might work for your own project. And being able to look up from your bench and look around, and also ask questions from people. I think people in the field are genuinely nice and generally nice. And I think we've talked about this before, hitting record, about being able to send out an email to a PI that you read their paper and they have something that, they can answer some question, or you can have an interesting conversation with them. Just go, just do it.
Yeah.
It's hard. Have your email reviewed by someone that you trust if you need it. I know I've done that. But just hit send and you never know. Worst case, you don't get an answer.
Exactly, yeah.
But you won't know if you haven't tried it.
Yeah, exactly. Yeah, the same applies to me. I was always, or am, quite shy in reaching out to people that I don't know. It's getting better right now. But I remember as a PhD student sending out an email to a PI in the field. I was, it was a challenge. And I was asking postdocs in the lab to review my emails before I hit the send button.
Yes, I always also felt like I needed to explain who I am and who I'm working with in order to kind of justify that email. And it's, I think people in general are really kind and nice. And as I mentioned, worst case, you won't get an answer. But if you don't try, you don't know. And sometimes most of the time, people will answer you and they will welcome your question or tell you, hey, I don't have the answer to this, but maybe you should talk to this person. And then the person is already CC'd. And most of my collaborations came out of these, serendipitous conversations. For example, we just submitted a paper to Science with a collaborator, and I've finished collecting, analyzing, and making the figures in 2015. And and this happened because I was at a conference, I presented a poster, someone stopped at my poster and said, oh, my roommate would love to talk to you. So we ended up talking, and it ended up being a collaboration. And it's a very interesting clinical project as well. And, it's 2022, it started in 2014, but, you never know who you get to talk to. And that poster discussion ended up a collaboration, but it also opened the door to my second postdoc.
Yeah.
And
that's a very interesting story that you were telling, but it also shows how you need to be patient sometimes.
Yes. Yes.
Because 2015 to 2022, this is a long time that sometimes people don't have, but you don't know how you will benefit from a certain contact that you make today in the future, in 7 years, for instance.
Exactly. And it got submitted about 2 weeks ago. And when I looked, I counted, we're 86 authors on this paper.
Yeah.
And I'm number 5, which I thought was really awesome. So I'm co-second.
Wow, that's great.
And it's amazing. I don't know if it's going to get through at Science or not, but it doesn't matter. I think it was just a fun project to work with. I did bred experiments in HEK cells, which were translated into patient cells, and we're reporting such an important description of mutations in G proteins, which I think is, again, really awesome to be part of, and you never know where a project might lead you, where just a conversation might lead you. So basically the take-home message is send that email. You never know.
Yeah, definitely. Fingers crossed for this manuscript.
Thank you. I'm very excited about it. So last but not least, top 3 aha moments that you had that shaped your trajectory as a scientist.
Yeah, I think the first one was when I realized how you can label proteins. That sounds very naive now, but I mean, imagine I studied pharmacy, where you have sometimes very, very old schedules, you learn very old topics, analytical chemistry, where you learn how to quantify whatever nickel ions in your sample. You have no clue about science, how this works, and then in the first month of your PhD, you realize that you can take the DNA of your receptor and insert the DNA of a fluorescent protein or attach it, and then the rest is done by the cell. This was very inspiring, and definitely one of the aha moments in my career thus far, how easy it can be. And from then on, I was always inspired or fascinated by different labeling techniques. Unnatural amino acid labeling is, for instance, something that I find very, very cool and very elegant. So that's one of the aha moments that I had. The second one, maybe when my first 96 well plate experiment worked out, because before I was always doing my experiments on a cover slip with cells. So you go there, you have your cells grown and transfected and grown on this cover slip. You go to the microscope with a perfusion system. You're basically trying to find the perfect cell by scrolling over this cover slip for several minutes, half an hour, whatever, and then you find this one cell, you perfuse it, and it's just washed away, and then you start from the scratch. But when you then instead use a microtiter plate and it works out, and I realized, oh wow, now I can basically generate 96 times the data in a much shorter time or the same time. This was very cool. And I realized how basically tools can speed up research. So like a practical moment that showed me how tools can speed up research. And the third one was, I think, these data that we generated and collected and then published about frizzles that really showed how they have at least 2 different ways of function, that they need to undergo conformational changes for this one path, but apparently these conformational changes are not required for acting through this, what is called the signalosome model, that, as I said in the beginning of the recording, that it depends on the phosphorylation and cross-linkage with Wnt co-receptors. That was showing to me, at least, that there's not only just black or white, but there can also be black and white maybe in nature, or something in between. And it doesn't care what you think about or what your opinion is. The data speaks for itself, and you have to interpret it and try to find the truth in it.
Agreed. And I think you made a great point about really being open-minded and thinking about how the data speaks for itself. And you may have a nice story in your head, but when the data is the data.
Yeah.
And I can also relate to how beautiful it is when you can run 96 well plates or 384 well plates. I remember when I started out and we used to quantify cyclic AMP inhibition and purify the labeled, tritiated cyclic AMP on columns, and you can do only, I think, 24 or 48 at a time, and it was column on column, and then you collect and you go to quantify your radioactivity, and then you come back the next morning and you realize after the third vial, the machine just stopped counting, and then you have to start all over again. And then we switched to the bread-based cyclic AMP biosensor, and then you can run 20 plates a day. You were tired after that, but you had enough data for the next month to be looking at.
Yeah, exactly. But you get adapted to this throughput so quickly that I notice myself being frustrated when one of the plates doesn't work.
Yes.
Although then I should always remind myself, man, look, a couple of years ago you were doing these kinds of experiments and like single cell assay, and you would have done that 96 times to have the same data output as now, and now it's just one plate that failed, so don't worry.
Exactly. But then again, I think it's the scientist in you, it's the curious scientist in you that wants to go faster and better and cheaper and sooner. And then you realize with all the data you generated, you don't have the computational background to actually batch analyze the data, which again gets you to a second frustrating point. So you need to find a collaborator who actually can do this quicker, and then you get your plots, with a touch of a button.
Yeah.
That is awesome. So, really last question, Hannes. What's next for you? You're now in your second postdoc. You mentioned you wanted to go for a permanent faculty position. Where do you see yourself? I know it's difficult to answer that because you never know where these positions are available. But if you had a preference, what would that look like?
Yeah. As you say, it's a difficult question because you don't know which positions might come up. But I'm definitely trying to follow this path and try to get funding to start my own lab. So there's different options in the EU, but also in Germany itself via the German Research Council to apply for money to start your own lab. These are different things that I will try, but of course I cannot risk just going this one path, but I also need to try to find faculty positions here. I definitely want to stay in Germany with my wife, my family, and I just need to see what the future brings. And I think especially for that, it's always very important, bringing us back to the point that we discussed before, networking and being open-minded and see what the future brings.
Agreed. And just send that email. You never know who you get an introduction to.
Yeah, exactly.
All right, super. Thank you so much for your time, Hannes. I really enjoyed our chat, and I wish you all the best. Obviously, keep in touch, and I'll talk to you soon.
Thank you so much. It's been a big pleasure.
Thank you. Thank you for joining us and listening to this Dr. GPCR podcast episode. I'd like to thank our team members, Attila Forres, Ines Pinero, and our newest Dr. GPCR proteges, Montserrat Avila Zozoya, and Nipuna Wirusingo. Welcome to the team, Monsi and Nipuna. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com, and please become a site member today at drgpcr.com. And until next time, stay safe.